This talk will discuss the analysis of microbial communities with the Pathway Tools software. Here's an overview of the Pathway Tools software. It is an extensive toolkit for enterprise pathway genome data management. It includes genome, pathway, and regulatory informatics, and its capabilities include search, visualization, and comparative analysis. It performs multiple computational inferences, including metabolic reconstruction and operon prediction. It has a number of omics data analysis capabilities. It can build quantitative metabolic flux models. It enables users to interactively edit the pathway genome databases that Pathway Tools creates, such as to add new gene functions and pathways. And it can run as both a desktop application and as a web server. Here's the overall data flow within Pathway Tools. We feed in as the input to the software an annotated metagenome or metagenome assembled genome. And then the pathologic component of Pathway Tools performs computational inferences such as predicting the metabolic pathways of the organism. It creates a new pathway genome database, which can then be operated on by these other components of Pathway Tools, including the interactive editing tools, the navigator for search, visualization, and omics data analysis, and the metaflux modeling tool. Now, the first use case I want to discuss with regard to the microbiome data is performing metabolic reconstruction from metagenome data. What this process does is to predict the metabolic network of an organism from an input annotated metagenome, by which I mean performing a qualitative metabolic reconstruction for the organism that predicts its reactome and their component metabolites of those reactions and predicts the metabolic pathways of the organism. Pathway Tools provides a batch mode that can be executed from the command line for high throughput. During this process, the software will calculate pathway abundances if the metagenome input data contains gene abundances. And the resulting pathway genome database that's created will enable information exploration of that information space via pathway, reaction, metabolite, and gene information pages that the software generates. The software supports 3 different input formats, namely GenBank format, GFF3 format, and our own format called pathologic format. Metabolic reconstruction proceeds in 2 different phases. In this example, we're going to be assuming that the input is in GenBank format, where for each gene in the organism or in the metagenome, we're providing the coordinates of the gene on its chromosome or contig. We're providing a gene name, EC number, and a product name. What the software does is it considers each protein in the organism and attempts to infer what, if any, reactions that protein catalyzes, and to import those reactions from our MetaPsych metabolic pathway database into the current pathway genome database. What the program will do is to match each protein function, such as tryptophan synthase subunit alpha, to a corresponding metapsych reaction or reactions. And it does that matching via 4 possible sources of information from the input file: an EC number, a gene ontology term, which is not present in this example input file, a gene name, such as TRPA, and an enzyme name, such as tryptophan synthase subunit alpha, or carbamate kinase. And one thing I want to emphasize about this problem of matching enzyme names is that enzyme names are uncontrolled terms, and they typically have a lot of other modifiers included, such as subunit information, cellular location, gene names that can be included with the product name. We have algorithms for honing in on the core enzyme activity to allow us to locate that, which can be looked up in MetaPsych, and thereby the program is not confused by all these additional words in the protein function. All right, so the output of the reactome inference phase is a list of inferred reactions for the metagenome that the enzymes in the metagenome can catalyze. The next phase of metabolic reconstruction is pathway inference. And I want to emphasize that the pathway inference occurs by recognizing pathways from our Metapsych database, which is a highly curated metabolic pathway and enzyme database. And it's really the most extensive microbial pathway database. So using other pathway databases for microbiome analysis are going to lead to less comprehensive results. What the software does is for each pathway in Metapsych, we compute a score expressing the likelihood that that pathway is present. And that score reflects many different types of information. For how many of the reactions in a given pathway are there enzymes present in the metagenome? And we ignore spontaneous reactions in the pathway. We consider whether a given pathway is outside its designated taxonomic range. For example, Metapsych has plant and mammalian pathways, and the scoring algorithm tries hard to avoid predicting those in bacterial genomes. We also consider whether enzymes are present for designated key reactions, which are key indicators of the presence of that pathway, because, for example, the Calvin cycle and the TCA cycle share most of their reactions in common. We need ways to differentiate that. And we've also implemented some special rules to distinguish variant pathways. For example, Metapsych contains 9 different variants of the TCA cycle, and we try to predict the correct variant. Now, I've mentioned Metapsych a number of times. Here's some statistics on Metapsych. Actually, the first line is the number of genomes in our overall biopsych database collection. Metapsych is our reference pathway database in biopsych. It contains curated pathways, reactions, and enzymes from thousands of organisms, but these are all experimentally determined pathways and reactions. Overall, Metapsych's information has been drawn from 68,000 publications. We have 2,937 pathways, about 17,000 reactions and metabolites, and we have mini review summaries written by our curators for different pathways and enzymes that add up to about 10,000 pages, textbook page equivalents. Now, the next use case I want to consider is what I call MAG informatics. The idea is Pathway Tools provides a number of ways of analyzing the metagenome assembled genomes that are assembled out of a metagenome. And the idea is that first, some software outside Pathway Tools partitions a metagenome into these separate genomes assembled out of the metagenome to reflect different members of the overall microbial community. The idea is that Pathway Tools can process each individual MAG, again provided as a GenBank format file or a GFF3 format file, to yield a set of pathway genome databases. And so the same metabolic reconstruction approach that I just described for metagenomes can be applied to MAGs. We yield a set of PGDBs, each database describing one MAG, and there are several benefits overall to this procedure. First of all, is we can apply the search and visualization and analysis tools to individual MAGs to explore those genomes in a lot of depth. Pathway Tools also supports searches across the entire community of PGDBs, the set of PGDBs for those MAGs. The software provides a number of comparative analysis tools, and it also provides tools for meta-omics analysis. And now let me talk about each one of these in turn. Now, overall, Pathway Tools has extremely broad capabilities, spanning genome informatics, pathway informatics, regulatory informatics, omics data analysis, and comparative analysis. And all these tools can be brought to bear on any PGDB created from a MAG. So for example, we have searches for genes, proteins, and RNAs, and a gene information page. We have a genome browser, blast search, multiple sequence alignment. We have multiple pathway related tools. We have searches for exploring the reactions, metabolites, and pathways present in the organism. We have information pages for pathways, metabolites, and reactions, etc. So all these different tools can be brought to bear to probe the contents of a given MAG. Now, I thought I would emphasize our search tools. We have a multi-search tools for genes, reactions, metabolites, and pathways. Here I'm showing the gene search tool. And first, I want to emphasize that this tool allows multi-criteria searches to the genes within a single PGDB or a set of PGDBs. For example, the user can search or filter by sequence length, by the subunits of a protein, or filter by whether we're searching for a protein or a tRNA or an RNA gene product. We can filter by protein copy number if that's known, by small molecule regulators or cofactors that the protein makes use of, etc. In addition, we can specify whether we want to search against a single PGDB, a single genome, or against a set of genomes. And here I've selected 8 different databases from those currently available from the Altered Shadler Flora, which is a small mouse microbiome community. And so in this case, we're going to search for the protein name tryptophan. We're going to search for all proteins that contain the word tryptophan across these 8 organisms. And I'll note that there are similar search tools available for reactions, metabolites, and pathways as well. The result of this search is a table that shows us in each organism which proteins did we find whose name includes the word tryptophan. So we can see that across these different members of the Altered Shadler Flora, there are different tryptophan-related enzymes present. This could have been a pathway search to find all the pathways whose name includes tryptophan. Could have been a metabolite search across this set of organisms to find all the metabolites present in these databases that have tryptophan in their name. Then I want to just mention the information pages we have available. There are protein information pages for viewing information about a particular protein within one of these databases. We have a pathway information page that depicts the pathway diagram, a metabolite information page, and there's a reaction information page as well. I'll quickly mention some of the omics data analysis tools that we have present. We have the ability to superimpose omics data on individual pathway diagrams, where in this case we're showing gene expression data. We can show gene expression data across a set of pathway diagrams. We can also show transcriptomics or metabolomics data on an individual metabolic map diagram for any one of the organisms in the community. In this diagram, we're showing pathways, metabolic pathways organized by pathway area, and we're highlighting the pathways with colors that reflect their expression levels. And when the user clicks on a given reaction, a pop-up window shows the gene expression levels for all genes catalyzing that reaction over the 6 time points shown here. Now we can actually create a multi-organism version of that diagram, or rather, for the 8 organisms in the Altered Shadler Flora, we've put all of their metabolic map diagrams together in one screen, and we've highlighted, in this case, rather than highlighting omics data, which can be done in the same way, we could highlight these diagrams with metatranscriptomics data. Instead here, we've highlighted the diagram to show what pathways are unique to each organism. So the pathways in orange are only found in this organism. These orange pathways are only found in this organism. Tools, we actually have a whole suite of comparative analysis tools, which I don't have time to talk about, but this one shows for each amino acid the pathway or pathways that synthesize that amino acid in each of, in this case, only 4 organisms fit on the slide, but we can see, for example, that this organism lacks arginine biosynthesis. A final tool I'll talk about is that given a set of PGDBs for MAGs, we have a tool called multi-organism root search, which lets us determine how microbiome organisms accomplish cooperative metabolic transformations that no individual organism can perform on its own. An example is the production of TMAO, which has been linked to higher cardiac risk, and it's been found that L-carnitine is the precursor metabolite from which TMAO is synthesized. And we use this as a sample problem to ask what organisms accomplish this transformation using what reactions. And so we had built on a single organism metabolic root search tool, present in Pathway Tools, that asks how a single organism can transform a starting metabolite to an ending metabolite. And what the software does is to compute a minimal cost path through the metabolic network from the starting metabolite to the ending metabolite. And what it's trying to minimize in this procedure is the number of reactions in the resulting path, the number of atoms that are lost through this series of reactions based on atom mapping information, and the number of reactions that are foreign to the organism, because the algorithm can import additional reactions from Metapsych to enable this transformation to occur. We have a tool that we published 2 years ago called multiorganism route search that, given a starting metabolite, an ending metabolite, and a set of pathway genome databases, will find an optimal multi-organism linear metabolic route from the starting to the ending metabolite. And we've extended the route search algorithm by adding one additional optimality criterion, which is to minimize what we call the number of organism switches. So in this case, the software has found a route from the starting compound to the ending compound that makes use of 5 different reactions. The colors indicate which organisms the reactions occur in. So the first 2 reactions occur in one organism, the next 2 reactions occur in a second organism, and the final reaction occurs in a third organism. The software will minimize the number of these switches from one organism to another. And our algorithm was able to find several solutions for production of TMAO from L-carnitine. So in summary, I've talked about the metabolic reconstruction process for both metagenome and MAGs that pathway tools can perform. I've talked about the notion that pathway tools can generate multiple pathway genome databases for multiple MAGs. It can search for genes, pathways, and metabolites across these databases. We can generate multi-organism metabolic map diagrams for omics data analysis, and we have a multi-organism metabolic route search tool. And I'll note that Pathway Tools is freely available to nonprofits. We charge a fee to commercial institutions. It runs on the 3 major platforms, and you can find out more at our website.